and welcome to the Amplifying Scientific Innovation podcast. I'm your host, Dr. Sophia Ononye, founder and CEO of the Sophia Consulting Firm, a life science marketing and communications consultancy that was established in New York City with the goal of amplifying scientific innovation. The goal of this podcast is to showcase scientific innovation stemming from global life science companies prior to and following COVID-19 from the perspectives of founders, CEOs, and other senior executives who are working on the development of transformative life-saving solutions for patients. My guest today is Mr. Daniel J. O'Connor, President, CEO, and Director of Oncosec Medical Incorporated, a biotechnology company pioneering new technologies to stimulate the body's immune system to target and attack cancer. Dan holds a Bachelor of Arts degree in English Language and Literature from Boston University, served the United States Marine Corps Officers Candidate School, and obtained his law degree from the Dickinson School of Law. Excitingly, I started out my career in corporate communications and investor relations at Oncosec in beautiful San Diego, and remain impressed by the company's innovative approach to developing cytokine-based intratumoral immunotherapies. I had the opportunity to meet Dan in person about a year ago at the Oncosec, New Jersey corporate headquarters following an introduction from the company's previous CFO and my longtime mentor and former boss, Mr. Richard Stansky. Welcome to the show, Dan. It's a pleasure to have you here.
Hey, thanks, Sophia. Really appreciate it. And it's great to see you again.
It's great to see you too. So we're going to go ahead and get started with what I believe is the most important question. What is your definition of scientific?
Yeah, so innovation it really starts with what you have, right? You take the existing learnings that you have and you extrapolate from them.
Mm-hmm.
And, from my point of view, it's extrapolation into something that is I'll relate it back to cancer immunotherapy. Yeah. Is then taking what you know and applying it into a kind of a hypothesis to say, how do we make this drug better, more efficacious, and how do we eliminate side effects? And so how do we make it better? Efficacy, as well as doing something that's nobody else is doing or can do. And doing that with your technology. And then, especially with respect to immunotherapy and cancer immunotherapy, is the underlying paradigm for cancer immunotherapy is not to take one form of side effects that you see with cytotoxic agents like chemoradiation and supplant them with a different type of side effects that you see with cancer immunotherapies. So innovation really is taking what we know and applying that and extending that into another area that is getting a drug to be more efficacious, but also eliminating the side effects and giving life to the underlying paradigm or the underlying big idea for cancer immunotherapy, which is using the body's own immune system and avoiding the ravages that we see with chemoradiation and other therapies.
Right. I really like how your answer sort of highlights the importance of efficacy and safety. That's really a unique response. And now I'm curious, what is your most notable accomplishment pre-CEO?
Before being a CEO at Oncosec, that's thank you for asking that question. Aside from convincing my wife to marry me many years ago, that was probably the most notable one. And thank God we're still married. No, I think that probably the thing I'm most proud of is the concept that relates to your last question, which is innovation.
Right.
I was at a company known as Avaxis. I conceived of the idea of using neoantigens and neoepitopes as a cancer immunotherapy with the goal of creating an immunotherapy and a target for an immunotherapy that was specific to the particular patient. So that, and we called it my immunotherapy neoepitopes. But that, was, when I look back, that was extremely innovative. It was honing in on a development that occurred with whole genomic sequencing and the ability to do that quickly. And then deploying that information and that, that sequencing into the setting of tumor biology and extrapolating what mutations exist in the tumor but not elsewhere in the body, and then targeting those mutations with their cancer immunotherapy. And so, that concept was a concept that I developed at Vaxis, I'm an inventor on that patent application. I don't know if it's been issued yet or not, but I look back and I know I appreciate the ability to have been able to work on that project and conceive of that idea.
Wow, that's really impressive. I mean, one would never know you have a law degree from the way you speak so well about science. I would think that you've been a scientist in the lab for years and years. That's really impressive.
Thanks. And, when I was a general counsel for, first for a CRO, and and the CRO world had the great benefit of seeing multiple different clients' projects. And I wanted to be a doctor growing up, but became a lawyer instead. But within the context of the CRO, I was able to really start to analyze, how the technologies that we were working with worked. I would participate in sponsor presentations where they would go through the technology, and that's really where I started to get an orientation towards immunotherapy and oncology. One of our clients was Medarex, and this is 20 plus years ago. And then at ImClone, actually I started to really study immunology because my oldest son, when he was a baby, he's 22 years old now, had food allergy, extreme food allergy. And my wife and I, being lawyers, do a lot of research. So started to try to put together an upstream idea for something to be a prophylactic against an inappropriate immune response to an antigen like peanut. And so started coming to the idea of using a live helmet, in that way. But at that time, I was able to really interact with the ImClone immunologists, who some of them were very gracious to teach me. And Erbitux, the ImClone's product, had a kind of an immunotherapeutic effect that was just starting to be kind of identified in the early days. So that's really where kind of the learning started. I had that desire early on, but through the good grace of some good immunologists earlier in my career, I was able to really start to get a scientific foundation. The other part that I did was I've studied. There's a great book out there that teaches immunology kind of all the way through. I'm really well versed in the first 2 chapters of this book. When I get into the deeper chapters, it gets harder for me. But it's and I can give to Jem the name of the author. I have it on my the tablet, and periodically reread those first couple of chapters because they're very helpful.
Wonderful. Well, reading is fundamental. It's really the best way that you can learn. So my next question is a bit more broad. Can you provide a top-line overview of innovation that is stemming from Oncosec, especially as it relates to immunotherapy and even possibly anti-infectives?
Yeah. So, so Oncosec uses a DNA transfer system, and it does so using electroporation. Electroporation, one of the things that was very attractive to me about that technology is, when you're dealing with cancer immunotherapy, you're always trying to figure out a way to interact with the immune response,
right.
To speak to the immune system, and and to communicate. So companies use adenoviruses because the immune system will gobble up a virus or a bacteria or an antibody, right? But those are all things that have physical structure. Right? An antibody is an antibody. It's got structure. A bacteria, virus, etc. Same thing. Electroporation, does that communication job by transferring DNA into a cell, but it does so without a physical thing like a bacteria or an antibody or a virus, right? It does it with energy, which is effervescent, right? It's, there's, once that pulse of energy, which is the electroporation, goes into a cell, into millions of cells, those cells become permeable and receive that DNA plasmid that we just previously deposited there. So that communication happens without a physical structure, which is one of the innovative aspects of the Oncosec technology that I like a lot. Technology that was used on the bench, but exported into the clinic. Because a very smart researcher said, we can use this system, go to a tumor with ostensibly a gene therapy, deliver something like IL-12, which is, I guess you could consider it the missing ingredient in tumor biology because it's a pro-inflammatory signaling cytokine, and tumors are usually immunologically suppressed, right? Mm-hmm. So reintegrating that, but doing it without a thing like that the body has to deal with, like a bacteria or a virus or an antibody, and then using that kind of nexus, that tumor biology, to communicate with the rest of the immune response, which again is, one of the things that I think really attracted me to the company is the innovative aspect of that, of taking an existing technology and applying it to, really kind of a gene transfer system, which has the ability to, going back to my first answer, right, deliver a efficacious system treatment to a patient without, complexing the patient's experience. In other words, not supplanting one form of side effects or another, because you're not using a bacteria that the body has to do something with, or that the body has a cytokine release syndrome response to, with hypotension associated with it. So, that's, again, I think one of the things that attracted me to the company is that ability to use this non-physical system to deploy genes, not just IL-12, but other genes of choice. And we historically have applied that to surface lesions, so skin and subs below the skin lesions. We've also now miniaturized the technology so we can put it through a catheter and get into the lung or the liver. Again, that was taking our learnings from the preclinical area where we had already miniaturized our electroporator, right, and kind of exporting that technology into get inside the body. With respect to COVID, same thing. It wasn't necessary. We know that that electroporation can be used as a way to create a vaccine, right? You get a sustained release of the antigen when you use it through electroporation, and other companies have shown that. So we know that technology can be used for a vaccine, but what we said was, could IL-12 play a role in enhancing the response to something like the glycoprotein that the NIH developed, for the so-called spike protein. Right. And so that's really kind of the next innovation as it relates to COVID, which is to say, hey, can IL-12, which we know a lot about, it's really, our company is founded on IL-12, can that play a role in enhancing a vaccine approach? And so we shared that information with others, and several others believe there's real potential there. And fortunately, for us, they said, we will take the program, we'll put the money into it, and if you will supply Tavo, that would be great. So anyway, that's kind of another way that we look to say, okay, how can we deploy this technology? Really, the impetus for doing that was to help, right? We have, a block long line in front of Walmart now, in almost every Walmart and, throughout the United States. And this is that's just a microcosm for what's going on, in so many other ways. And so we wanted to help. And, that's where we said, can IL-12 play a role? And the answer from the immunologist was it could potentially play a very good role in creating a more efficacious vaccine.
I think that's brilliant, and it really ties into my next couple of questions, because I was going to ask you how has COVID-19 changed the way you work internally? I was going to follow it up with how your corporate social responsibility efforts have been changed as a result of that. But I think you responded quite precisely around that. Do you have anything else that you want to add?
No, COVID, I think, has changed the way we work. And I can say that before what's happened, I liked being in the office and I wanted everybody to be there. It was a bit of an old school approach, I'll admit it. But I think, old dogs, new tricks. It can be done. I like, I've learned that you can actually be very effective working from home. I think the one thing that we have to get back to is the human exchange, because so much of what we do, or a lot of what we do, happens with not just the video conferencing interaction, but when you're sitting in a room 8 hours a day or in an office with the same people, a lot of things come from that. It's an intangible that we do have to get back to. But I think for me, when someone says, hey, can I work from home in the future, I'll be a lot more receptive to the idea than I have historically been.
Yeah, well, I think positively, be open to new ideas. I think that's sort of what I take away from our conversation so far. Now, my next question for you is, thinking broadly, what do you think are some key consideration factors that would help to sustain innovation in the life science industry?
Yeah, I think we need to continue to use the things we know and bring them forward into kind of a next version of those things,
Yeah.
or better use of those things. But also, I feel like we need to really start to hone in on what I'll call sustainability of a drug. We have systems that we built that are complicated and expensive. And I question whether or not they'll actually ever be used or used, right, be able to be reaching a lot of people. I feel like we need to start to continue, or we need to think about drugs that can be, what I call sustainable, meaning that, they can be used, they can be cost effective, and they can deliver good results for patients. I feel like as we continue to think about healthcare costs and how to address that as a topic, one of the areas I think we need to continue to think about as manufacturers is, what are the costs associated with the product, just the product itself. The clinical development costs are phase 1, phase 2, phase 3. Those are fixed in a way, unless there's a change in how that happens. But the other costs are, what does it cost to make your drug? And and what are the side effects that are required to manage your drug, right? What other treatment, once you get your drug, are you going to have to get to manage your drug? It's going to be hospitalization, et cetera. So I think, when we think about innovation, we have to try to fold in the idea of drugs that are sustainable, meaning that their costs to make the drugs themselves are within reach and are not going to be extremely expensive, such that they're not going to be able to be widely deployed.
Right. No, that makes absolute sense. Now, my next question is fairly tied into the previous one. So is there any company, including yours, or technology that you're currently excited about, and why?
Yeah, ours is really, it's a self-serving short answer, but it's true. I like our technology a lot. I think, again, it kind of gets back to that one answer, which is, we don't want to develop an immunotherapy that supplants one form of side effects for another. Right? The whole idea is, let's use our body's own immune system. And, I really like the idea of using energy to cause gene transfer, as opposed to a physical structure to communicate with the immune response. I think it's got a lot of application. I think it's just in its nascent form, right? It's got a lot. It's got a long way to go, and it's got a lot of development potential. I think about getting into a liver lesion with something like IL-12 or CXCL9 or any other number of immunologically relevant genes that can be transferred to a tumor cell to get that cell making that gene, IL-12, or maybe CTLA-4. Right, avoiding the entire systemic toxicity associated with some drugs, and delivering them directly to the tumor. I think about the same type of gene transfer for lymphatic tissue, and playing within the cycle of the entire immune response. So I think electroporation itself, while some may look at it and say, oh, it's kind of old school, or it's low tech in a way, it's really not. I think it's very simple and kind of ingenious. And I think it's got a lot of applications that have not been tapped into. The other technology I do like, I think, is the cell therapies. I think the work that's being done there is tremendous. And I think, it relates back to that earlier point, was sustainable drug versus not sustainable drug. But, I think as we go on, as we continue to consider cell therapies, we have to figure out ways to make them in a way that's not going to be super expensive and it'll never get utilized, kind of thing. So, but I do think the cell therapy area is an important area.
Wonderful. I want to piggyback on what you said earlier. I completely agree with you about innovation. Electroporation, even if it's an old technology, as long as we can find new ways to repurpose what we know, that is innovative in its own way. So, and I think that's an important point that you
Thanks. Thank you.
Now thinking, imagine this, there's a new biotech CEO, they start next Monday. How would your advice for them be different today versus prior to COVID-19?
Yeah, don't do it. No, just kidding. Just kidding. That was a joke. No, I don't think the advice would change, pre and post COVID. Honestly, there's plenty of advice for a biotech CEO. You could probably write a book on it. But I don't think it's pre and post COVID. I think, yeah, so it's more of a kind of a universal advice, and there's a lot of things to think about. I mean, I think the number one thing to think about for a biotech CEO is don't be afraid. Fear paralyzes, and you have to be bold, and you have to, do a lot of biotech by definition is doing things that people don't want to do or can't do, or those kind of things. It's the courageous that go out there, but it's scary and it can be daunting. And when fear creeps in, you just have to keep pushing it down and keep moving.
Wonderful. So any other commentary or thoughts? The floor is completely yours.
Oh no! I really appreciate your interviewing me. Like I said, it was great to see you again. I'm impressed by your, like I said, pushing fear down and going for it. You're doing that. I think that's excellent. And encourage you to keep going. I guess probably that's it. I want to wish you success, and because I know when you do something and start something, it can be scary, but you're doing it. So great job, and keep it up.
Thank you. The only thing to fear is fear itself.
Yeah, that's good. That's somebody said that one. I'm sure. It was very appropriate. Actually, it's a very true statement.
All right. Well, thank you, Dan.
Thanks.
You have a good rest of your day.
You too. Thanks for speaking with me. Take care.
Thank you. Bye.
See ya.